Okay, thank you very much for the introduction. As you have all heard, my name is Dr. Mafa. I am a biochemist. My research area particularly focuses on the biomass degradation using glycosyl hydrolase enzymes. And for those of you who will be interested in that part of research, I have my ORCID number on the screen for a number of publications that were produced from that work. However, I also have a very keen interest in understanding how carbohydrates play an important role in defense responses against biotic or abiotic agent. So the work that I'm going to present here is done by a master's student, which I'm co-supervising, and his name is Mr. Jeremiah Hlahla, and the main supervisor is Dr. Moloi. So just to give you the background of glycine max, which throughout the presentation I will call edamame, is actually a vegetable soybean, and its origin is from East Asia, particularly in China, Japan, Taiwan, as well as Korea. So this cultivar is actually this plant is actually very old. It has been documented to exist for more than 2,000 years. And it was called with different names in different places. And there's one paper that cited that the name edamame actually is more, it was derived in Japan. And due to its nutrition value, edamame was introduced in North America, South America, as well as Europe. And recently, it is introduced in Africa. However, there's a need to research the conditions and the cultivars that would be best suited for the Southern African region, which I'm based. And that's why we're doing the study that we do. So unlike the grain soybean, which is generally used for oil production and protein, edamame is mainly used as a food source. Mostly in Asia, you can find it as a snack, whereby it's boiled in salty water, and you just eat the seed in that case. However, the second part is where you can eat it as a vegetable, and it's prepared as a stir fry or in vegetable stew or even a soup. So what makes edamame different from the normal grain soybean is that at maturity, it has a dark green color, and it also has larger seed size and soft texture of the pod, and it has a sweet and less beany flavor. And this plant has lots of health benefits due to its high content of vitamins, minerals, as well as a number of polyphenol like isoflavonoids. And we're not going to focus on its medicinal properties, but we want to understand its drought tolerance responses in this case. So looking at the developmental stage of these plants, we noted that there are two developmental stages that have been documented in literature. You have the vegetative stage, and you also have the reproductive stage. The vegetative stage consists of about 6 stages, where you have the emergence of the seed after germination up until you have the 6th node just before flowering stage. With regards to reproduction, which is one of the most important stages of the plant development, is you have 8 stages, and they are denoted R1 up to R8. R1 just means that you have the beginning of the flowering stage, and R2 is when the plants are fully blossomed. And then R6 is generally the stage where the plant is harvested. And we're interested in these 2 stages. So we harvested the leaves of the plant from R2 and R6 stages, as you will see when I discuss my methods and materials and the results later on. The reason why a number of researchers have suggested that plants should be harvested at R6 stage is because it releases ascorbic acid at R7 stage, and this acid kind of turns the color of the pod yellowish, and you also start to lose a number of nutritional value in the seeds. So R stage 6 is very important because that's where you still have maximum nutrition in the plant. So, like I said, the physiological as well as chemical properties of edamame have been well documented previously. It was demonstrated that most of the cultivars accumulate their lipids at stage R5 up until R6, and immediately after R6, the lipids will decline. But during R6, you also have a accumulation of proteins. And this is important because you get these proteins just before harvest, which is R6. And you also get a slightly increase of glucose from the developmental stages of R1 up until R6. However, the sucrose, which is one of the most important molecule for the seeds, which gives them their sweet flavor, is actually depleted after stage R6, particularly in stage R7 and 8. That's where you start to form very recalcitrant oligosaccharides called raffinose, as well as starches. And these, as you can see here on the left, let me get my pen first. Yes, as you can see, we have the raffinose there on your right bottom screen, and you also have the starches, which are oligosaccharides. And these oligosaccharides, they cause discomfort in higher content if you eat the edamame that contains them. So the seed development as well as the storage information is well documented with regards to that. However, we want to study the edamame in the South African context because the World Health Organization, not the Health World Organization, but the Food Agricultural Organization of the United Nations actually issued a report in 2018 that highlighted that 1 in 9 people around the world are actually suffering from undernutrition. And one of the countries or continents that reflect was Africa. So hence, it is interesting that edamame is introduced in South Africa due to its affordable price and as well as nutritional content. However, there is a lack of information on the responses to drought, and particularly in South Africa, 2016 up until 2019, we were hit by severe drought. So it will be interesting to see how these plants respond in the South African climate, because in United States and in the Eastern Asian countries, a lot of research has been done, and they understand how to cultivate the plant. So the aim of our study was to investigate the photosynthetic efficiency of and the carbohydrate osmolyte response of 6 edamame under greenhouse conditions of drought. So to discuss the methods and material, we first had the 6 cultivar, which were denoted UVE7, 8, 14, 17, AGS 354, AGS 249, and they were germinated on a tray using a hydromix. After germinating, we then transplanted the plants into the pots that has 10-liter capacity, containing 10 kg soil, and then these were placed they were grown in the glass house at 25 degrees during the day and 18 degrees at night. The plants were then watered to soil capacity, which was 100% until a third trifoliate stage. And then thereafter, we then started inducing the drought stress in the plants by supplying them with 30% water capacity, which was 0.48 liters compared to 1.6 liters of the control. And then after that, the plants were then, the leaf samples were then harvested at R2 and R6 reproductive stage for biochemical analysis. For chlorophyll fluorescence measurements, we then used a technique suggested by Parikh et al. And in his study of 2019. And this method is actually a non-invasive method where we use a portable probe that has the chlorophyll fluorometer, and we measured using the light energy that was set at 3,000 micromole per square meters per second. And then before we actually measure the sample, we first clipped the probe on the leaf that was fully expanded, allowed it for 30 minutes, and then after we took the measurements in the dark. And equation 1 was then used to determine the photosynthetic capacity of photosystem 2 and photosystem 1, as well as the total photosynthetic capacity was then, the readings were received from the machine as it did automatic calculations for us. As well as the carbon dioxide conductance was also performed using the probe that is non-invasive, where we also used the parameters that were given by the machine, and as you will see later on the results. So we then went ahead and quantified the pigments in the samples, still using the methods that were suggested by Parikh et al. 2019. Firstly, we then weighed 100 milligrams of leaf tissue. And then after that, we extracted using 80% chilled acetone, centrifuged the samples. Then we got the supernatant. The supernatant was then used as the measure for chlorophyll A, chlorophyll B, as well as carotenoids at the mentioned wavelength on the screen. We then determined the amounts of chlorophyll pigments in milligrams per mil, as you can see from equation 2, 3, and 4. So after that, we then continued, and then we measured the soluble carbohydrates, and we were actually interested in knowing whether we will get maybe glucose, some of the oligosaccharides or disaccharides. Firstly, we then used 80% ethanol as to extract the metabolites, and then in the supernatant, after centrifuging, we then added 40 milligrams of charged charcoal, and this charcoal was then centrifuged, and then the clear supernatant, which was transparent, was then used to determine the sugar content using the Megazyme kits, which are commercially available. We used the glucose kit, the Trehalose kit, and the starch kit. There is a paper of another research group in South Africa from the University of Cape Town, which have actually demonstrated that the abundance of glucose, Trehalose, as well as starch can be used as the marker for defense responses against drought or heat stress. Lastly, we then determine the yield parameters by actually harvesting the pod with the hand at the maturity stage, which was stage R8. And now I'm about to show you the results that we have received. As you can see, we have two tables that demonstrate the chlorophyll fluorescence parameters. The values represents the mean, and we had about 5 replicates per sample or per cultivar. And we also performed the statistical analysis using a two-way ANOVA. And as you can see on the right of the columns, you have, of the tables, you have the arrows. The green arrow shows that there was a decrease in total photosynthetic capacity of the cultivars, while the red arrow shows the increase chlorophyll fluorescence capacity. So if, for instance, if you look at the AGS 354, there was a decrease when you compare the plants that were induced with 30% drought and the control with regards to the total fluorescence parameters. However, with UV8, UV14, and UV17, there was a higher fluorescence capacity in the drought-induced cultivars. When you look at the plants that were actually harvested during the pot filling stage, we also saw a little bit of a variation in terms of responses. Now the AGS 249, which didn't show much of a difference during the flowering stage, showed a significant decrease in photosynthetic capacity, and the AG S354, which was decreasing during the flowering stage, started to increase. Of interest is this cultivar UVE14. Its photosynthetic capacity in both cases was still high, while UV8 did not show any significant difference. When we then looked at the pigment content with regards to whether it can tell us what is happening with regards to the fact that there was a decrease in photosynthetic capacity in some cultivars and there was an increase in others. We also saw a little bit a trend that was expected, because if you compare the control, which received 100% capacity water, and then the drought-induced, which only received 30% capacity, we started to see that chlorophyll B content was actually decreasing. However, chlorophyll A and carotenoids content were pretty much stable. And if you compare the flowering stage with regards to the pot filling stage samples, then you can also see that there was now an increase in chlorophyll B content in almost all the samples that were decreasing, except for one sample, which is UV14, which did not show much of a decrease. But when you compare this sample with the sample in flowering stage, there isn't significant difference in terms of shifting. And I'll explain later why I'm bringing your attention to cultivar UV14 later on. Then looking at the stomatal conductance, the stomatal conductance gave us the results that we expected, because generally drought is associated with less water. The plants will then induce the abscisic acid, which will then close the stomata. And we can see that during the flowering stage, all the control had higher stomatal conductance, and we had about 50% reduction in stomatal conductance. However, when you compare the stomatal conductance of the plants during the pot filling stage, you can start to see that UV14 was recovering, had about 20% loss of stomatal conductance relative to control. Also, when you compare AGS 429, it also had less stomatal conductance in drought-induced, but it was much better compared to the flowering stage. So it tells us, this tells us that during the pot filling, you required more photosynthesis because the plants were starting to pack metabolites into the seeds, and therefore it makes sense why there is an upregulation of photosynthesis, even though it's still not more than the control, but that increase was an indication to us that at least there is something that is happening that we are expecting. So regarding the sugar metabolites, we then looked at glucose concentration in the plants that were exposed to drought stress. So the glucose really varied between the pot filling stages as well as flowering stages, like other parameters, except that if you look at glucose now, you will notice that glucose was mostly significantly different in reduced in other samples, but for sample like a UVE7, there was no significant difference between the control and the treatment. And also between UVE17, you also had like only about 20% loss of glucose content. And when you go to pot filling stage, there was no significant difference in almost three cultivars, which is AGS 354, AGS 249, UV7. And even though there was reduction in the remaining cultivars, it was not more than 60%, which kind of showed us that the plants were somehow responding positively towards drought induction. Then we looked at the trehalose, which is the molecule that is proposed that is only induced during abiotic stress. Apparently, this molecule, there's a number of papers that have demonstrated that it is absent in plants that are grown under normal or optimal conditions. However, in our case, during the flowering stage, we could see that we still had a number of cultivars that had high trehalose as the control, but it showed that UV14 had the highest induced trehalose relative to its own control, even though its amounts were lower relative to other cultivars, as well as UV8 had the high insignificant difference between the control and the treatment. However, when you look at the pot filling stage, there was a little bit of trehalose decrease in other cultivars that showed trehalose increase, and there was more of this trehalose being induced, which supports the view that trehalose is mainly induced during abiotic stress, and it assists in terms of homeostasis of sugar osmolytes in the plant. So looking at the sucrose, which is also one of the most important molecule for this edamame plants, as I have said, it plays role in also in terms of flavoring the seeds of the plant. We noted that when we tested the samples for flowering stage, we couldn't get any sucrose with the kits that we were using. And at the moment, the student is trying to repeat this study so that we can double check whether it was just a mistake or it was exactly what happened. Because when we performed the same experiment but with pot filling samples, you can clearly see that sucrose is highly induced in the drought in the drought stressed plants, while in the control it is present, but it is very low. And this was a very significant increase. And sucrose is also one sugar metabolite that is also recommended or suggested that it plays a very important role in the homeostasis of osmolytes during abiotic stresses such as drought or heat stress. So lastly, with regards to the carbohydrate metabolites, we then looked at the starch content of each plants during this flowering stage. And we noted that, again, UV14 had induced starch content. UV8 had induced starch content that was significant. And others, as well as UV7 had induced starch content, but there was no significant difference in other samples. So only 3 had a very significant induction of starch content. And this also explains why we saw high amounts of trehalose as well as the glucose metabolites in UV14 during flowering stage. However, when you look at the pot filling stage now, only AGS 429 had the highly induced starch content, while others reduced their starch content. Again, we also had high starch content in UVE 17. And with regards to the control here, we couldn't get much values that were making sense. So just to wrap up this section of the results, we then looked at the yield. So for the yield, what we have done is we allow the plants to grow up until the last developmental stage, which is R8. And at that point, we then calculated the agronomical parameters of yield, such as the total pods per plant, the total seed per plant, total seed mass per plant, total flat pods. Then the flat pods means that the pods did not have any seeds in them. And then we also looked at how many pods that were shattered and what was the mass of seeds per 100 count. And then we looked at the stem length as well as the branch per plant. As you can see, again, the green arrow shows the decrease in the number as compared to highly increased seed per plant. You can see, for instance, we had UV14 having 48 seeds per plant. Again, this demonstrates that the high photosynthetic efficiency, high sugar metabolites accumulation in this cultivar, as I've been flagging it throughout the presentation, resulted in it having the highest amount of seeds. However, when you then look at these parameters of control and you compare them to the drought stress plants, then you still see that UV14 had the highest amount of seed per plant. However, when you look at the mass of 100 seeds, you then also see that UV14 had the highest mass for the plants that were exposed to drought stress. Then now, in summary, we can say that photosystem 2 was relatively stable in all the cultivars during the flowering and the pot filling stage, but photosystem 1 was actually increased in UVE8, 14, and 7 during flowering stage. The pigment content data demonstrated that chlorophyll A, carotenoids were stable in the control and in the drought cultivars, which was very important because it shows that the photosynthesis is still, was still at its peak. And except for that, chlorophyll B was decreasing in drought-induced plants at flowering stage. And one of the most important findings were that at flowering stage, the gases exchange was actually half the compared to the control, but it then increased about to 75% increment. And therefore, that can explain why we ended up having the seeds in the drought-induced plants, because it shows that the gases exchange was happening and we had photosynthesis happening at highest level during the pot filling stage. And then I would just like to conclude by saying that we suggest that UV E14 as well as AGS 429 can be used for breeding studies in South Africa to introduce this cultivar for cultivating edamame. I would like to thank my collaborator, which is Dr. Moloi, and Jeremiah Hlahla, who has performed all the experiments and who has done the hard work for us. And I would also like to thank the funders, which is NRF, and then the CRF fund and the University of Free State for allowing us to do this work. And then lastly, I would like to thank STEM for giving us the opportunity. Thank you very much.